Hello everyone, good morning. Thank you so much for joining us today. And actually, my lecture is going to be a little different. I want to bring you to the lab with me. I want to show you what we do in terms of clinical analysis of samples that come to us for microbiome studies, and also a little bit more of how we do research as well. Right, so I will walk you through to the entire process from sample prep to the results, even showing you a few reports that sometimes we release that may help you in the future to understand how this microbiome is actually reported to you and why it's important. Right, so actually I divide this lecture in two blocks. So the first part, we are talking about the clinical perspective of the microbiome assessment. And with that, I will show you how we isolate and identify potential microbes that are disruptor of the small bowel microbiome or even fecal microbiome, right? And then later on, we will walk you through a little bit of more broad range perspective and research likely type of techniques that we apply in the lab that usually we use high throughput technologies where we do DNA sequencing and pathway analysis to understand these microbiomes. Right, but first, with a single slide, I want to show you why actually we had to develop all these techniques and why we need to employ these pipelines to study these microbes. So Dr. Mathur just showed you very beautifully what is the microbiome, right? So the microbiome is a mixture of different types of microbes all together interacting. So we are talking about bacteria, we are talking about fungi, we are talking about archaea, even parasites that interact together in a specific site. And all these microbes, with a few exceptions, they have one thing in common. They're very small, right? So we cannot see them with the naked eye. And because of that, it's very difficult to isolate and identify these microbes. So that's why we need to do more research. We need to create techniques in order to do that, right? Including the clinical side. So we don't just get a sample in the lab and give you right away a result of what is there. So we need to employ some techniques, and this is exactly what I'm going to show to you today. So basically, we have one goal in the clinical lab, and one goal only: isolate and identify microbes. We want to know if these microbes are disruptors of your small bowel microbiome or fecal microbiome or any microbiome that we are studying. And there is only one way to do that: by isolating them, by identifying them with specific techniques that can be a biochemical test, can be a protein analysis, it can be a DNA sequencing, it doesn't matter. We need to identify this specific pathogen, right? And then, because of that, is when we can release the report and understand better how these microbes are interacting with your normal flora, with your microbiome. So first things first, I want to walk you through since the sample collection throughout the microbe identification. So we receive samples in the lab. What we do first, we give a quick look in fresh samples. We want to see it, what's going on under the microscope, right? So in order to do that, we need to employ a few techniques, right? They are not exactly easy to visualize, but we can do a few things to do that. And one of the things that we do is a staining technique. Some of these microbes, they can be stained. You can use a mixture of colors so you can see them on the microscope, and that actually gives you a little bit of information of who they are, right? So we employ grain stain technique, and we observe them on the microscope, and then that give us a direction what should we do next in terms of growing them and identifying. Right, so during these staining techniques, usually we see two types of colors on the microscope. We see what we call gram-negative microbes, and these are very important. I bet you heard a lot about them before. So these gram-negative bacteria, they're usually involved with infectious disease. They're usually involved with conditions such as small intestinal bacterial overgrowth, and we can actually see them on the microscope. So you can observe that they have red colors, they have a specific shape. In this case, it's a bacilli shape, right? And then on the other side, we can also see microbes that stain in purple. We call these gram-positive bacteria because they're purple, right? And then they also have different shape. So in this case, they're cocci shape. Sometimes they live together as a pair. Sometimes they live together like a grape, like grapes, right? So, and then this is the first step in what we should do in the lab in order to define the next steps. And then during this step, as I just mentioned, as we are visualizing your samples on the microscope, we can already take a few information about these microbes. One of the things that we can't see it is how they look like. And this is very important to us. It's like understanding their shape, if they have a bacilli shape, like this one, if they have a cocci shape, like this one, or the Y shape, we will define what to do next. And a lot of these shapes are associated with specific pathogens and give us some information like, okay, it's potentially a Escherichia coli. If it's a bacilli, that I see color as red on the microscope, depending from which site we collect these samples, right? And then we move on for the next step. So after we visualize these samples, we still have a lot to do. That's just the beginning, right? And then one of the things that we need to do is search for other microbes present in a specific sample. So we are not only, again, in a microbiome environment, only to bacteria. Although most of the research that we do these days is mostly on the bacterial side, let's remember that these microbiome have other types of microbes as well, such as fungi. And we see them all the time, even under the microscope. So we see fungi such as Aspergillus here, and this is a beautiful structures that we can see under the microscope, which can almost already tell you the species that is dominating in a specific environment. Or in this case, Candida albicans. And Candida albicans is a very important microbe, especially in the small bowel. It's one of the most important ones and associated with patients with small intestinal fungal overgrowth. Right? So then, after we search and we can see what is the majority of the microbes in a specific site, we can do what we do the best in the lab. We need to culture these microbes for further identification. And culturing these microbes involved a lot of steps, right? So the first step we already took. We know how they look like, we know their shape, so we know what to do next. How we know what to do next? Because depending on their shape, depending on their color in a grand staining, depending if they are fungi or if they are bacteria, I can select specific growth medium to make them grow in the lab in a way that I now can see them. So the goal during this step is grow these microbes enough to a point that I can see them and I can apply specific tests for identifications, right? So then I have a few examples of growth medium that we use in the lab, including the McConkey agar that we will talk about it, that we usually use to diagnose patients with small intestinal bacterial overgrowth. We also use the blood agar, where we can grow more fastidious microorganisms, some of them also disruptors of the microbiome, such as Clostridium difficile, such as, although we will not speak about this today, but Lactobacillus potentially as well. And we have another agar over here that is mostly for fungi, that is the Saburo agar, that we can grow Candida albicans, right? So after receiving the samples, we took a look. Now let them grow on this specific medium and take into consideration a lot of characteristics of these microbes. If they like oxygen or not to grow, if they need specific nutrients to grow, and we do that, right? So after we got the samples, we plate them on those agars, right, like you see in this picture. And then we incubate them in this incubator where we can control the temperature, we control the pressure, and we can control the exposure to oxygen, right? So we have plates that are completely exposed to oxygen, so we can grow aerobes, like Dr. Mathur just mentioned to you, like we have aerobes growing in the small bowel or stool samples as well. Or you can grow them in these specific jars where we can remove the oxygen content from the environment. And those bacteria that don't like oxygen will start to grow as well. So we do both at the same time, right? And then right after, we'll wait a little bit. Sometimes we wait 18 hours, sometimes we wait 24 hours, sometimes we need to wait even 48 hours, and for a few microorganisms, up to 7 days, right? So, and then what we see right after that, if there is a microbe and with a potential to grow in this specific medium, you're gonna see plates like this one. And this is a very nice example that we have from patients in the lab, right? So we use multiple agar with the same samples from the same patients, and all of these dots over here, they are bacterial colonies. We call them colonies. They came from a single microorganism that was present in a specific sample. So they grew that now I can see it. I don't need anymore a microscope in order to, okay, like there is a microbe growing at this specific site. And they have different colors and different shape, and I will show a few more pictures to you, but all of this information that comes from this step can also help us to identify these microbes. These colors means a lot to us. So let's suppose this agar in the middle, we call hectoine agar. The hectoine agar is an amazing source of nutrients for pathogens, such as Salmonella, that perhaps you already heard about, Shigella, Escherichia coli. And depending on the color that they show on the agar, it gives me already a hint of who they are. So if it's Escherichia coli, it's gonna grow orange. If it's a Salmonella, it's gonna grow green with a black spot in the middle. And that black spot in the middle also means a lot to us, and I will show you why, right? But at the end of the day, this step will give me a lot of information about what is the potential species. I cannot tell for sure yet which one, but now I have enough samples to do and to go to the next steps. But I wanna go over a few of the agars that we use in the lab because we use them to isolate microbes that are disruptors of the small bowel microbiome, of conditions that we would talk about today. We are talking about IBS, post-infectious IBS, we're talking about SIBO as well, and all these microbes can be studied in the lab. And one of the agars that I want to talk about is the McConkey agar that I mentioned a little bit. This is a very important step, and this is a step that we perform with every single sample that comes from the Reimagine study, where we need to define if a patient has small intestinal bacterial overgrowth. How we do that? We culture the samples, the asprays that we collect from the small bowel, such as Dr. Rizai showed to you how we do it, right? And we wait them grow to about 24, sometimes 48 hours. And then we see these little colonies. These are plates from patients, real patients that we have in the lab. And we see these colonies growing. It's like I told you, one colony came from one single microbe. And what we do next, we count them. We count one by one. And then in this case, I count 1, 2, 3. Of course, we have some technology to do that for us when the amount is excessive, but we count them. And then when we realize that this number is higher than 1,000 colonies per ml of aspray that we collected, that sample is positive for SIBO. So this is how we diagnose SIBO in the lab, right? But more than that, during this stage, we can also take a look closer to these colonies, and you can see how different they are. So you see that some of them are pink, some of them are yellow. Some of them glow more than other ones. Some of them are perfect circles, some of them a little bit spreading on the median, right? And this is also give us information about the potential species. So 2 of the species, the major species associated with SIBO, that is Escherichia coli and Klebsiella pneumoniae, they grow pink, like these ones on the McConkey agar, because they use lactose as a source of sugar. And this is very important to us to know that they are using lactose, and they have all the genetic machinery to break down lactose. They do more than that. I will show that to you. And then we have other microbes that don't like lactose. They don't use it. They don't have the machinery. And actually, in terms of pathogens, those that don't like lactose, they are the ones that we are concerned, right? So usually Salmonella, Shigella, they don't use lactose. They will show in the agar as these little yellow colonies. But then if we have an overgrowth of Salmonella and Shigella in your samples, we are not thinking anymore perhaps of SIBO. We are thinking about something that needs to be treated with antibiotics, right? Perhaps infection. But in this case, at least in the samples that come from the Reimagine study, we are searching for those associated with SIBO, right? So this is one step. This is just one side of the story. We have other disruptors of the small bowel microbiome as well. So one of the other one that we can see it and we can grow is Clostridium difficile and Lactobacillus. So these ones, they are more fastidious types of microorganisms. They need more. They need nutrients that usual agars don't have it. And this is part of how and why they are so virulent and so why they are so good as pathogens. So we need to actually use blood to grow them. They like that. So we create these blood agars where there's a mixture of nutrients with actual blood. And that's the only way that these microbes can grow in vitro. And these microbes also, in the blood agar like this, in a culture like this, you can see a lot about of who they can potentially be, right? The shape of the colonies, and even if they have the ability of breaking down the blood, right? And create this shadow around the colonies. We call this hemolysis, right? So the presence of hemolysis can already tell you if some of these microbes have these enzymes that can break down blood, right? Which is very important for their virulence. And we can see that on the agar as well. So for the Reimagine studies, for the perspective on research, we mostly work with these two agars, McConkey agar and blood agar. And the third one that we do, that we are recently doing more research and understanding, is the one that I mentioned to you before, that is the Saburo agar to grow fungi. So it's mostly Candida albicans, but we have other types of fungi, that yeast, that can grow in this agar. But we do that in the lab as well, where these agars are very unique because they have components that inhibit the growth of bacteria. And only fungi can grow in it, right? We develop these components that can inhibit cocci, can inhibit bacilli, although in some cases these bacteria can overcome these inhibitors, but there are a few exceptions. But the goal of this agar is to see if there is an overgrowth of fungi in a specific sample, and we see that. And more than that, what we do as well, that for fungi specifically, and only for fungi, there is one agar specifically for Candida that we can employ and use it to do already identification without any biochemical test, any DNA sequencing test. We can already tell the Candida species that are overgrowing a specific sample just by the color that you see in this agar, right? So then we have, in this case, this is the chrome agar, where it only grows Candida, right? And depending on the color, you can see that is a different types of Candida, right? So we have here Candida auris, that is the light blue color, and if it's a little different shape of the colony, a little lighter blue, is Candida albicans, that is the one that we see the most in the small bowel, right? And then if it's dark purple, it's Candida tropicalis. And then if it's pink, it's Candida krusei. And then if it's another shade of pink, but smaller colony, it's Candida glabrata, another important player in the small bowel. So this is one of the exceptions that we have, where actually you can already tell the species just by looking at this, the shape and the color of the colonies, but not for bacteria. For bacteria, like I mentioned before, we need to grow them, we see them, we have a few informations, okay, they are gram-negatives, they are gram-positives, so who they might be, they grew orange in the agar, they grew pink in the agar, or they might be Escherichia coli, they might be Klebsiella, but I cannot tell yet for sure. I need to do more. So what can we do in the lab in order to identify these microbes? And so far, I'm only working with you, walking through the clinical part. I didn't even start what we do for research, right? So we are trying to isolate specific disruptors of the small bowel. So then what we do biochemical tests. So after we isolate those little colonies, and one colony came from a single microbe, remember, we can apply a huge range of biochemical tests with these specific colonies to see what these microbes are capable to do. So let's suppose we have here a range of carbohydrate tests where we can see which carbohydrate they can ferment. And that tells me potentially of who they are, right? So if they can ferment only glucose, if they can ferment lactose, if they can ferment other types of carbohydrate as well. But during these tests, if they ferment some of this carbohydrate, can these microbes produce gas? This is another feature that we need to check in these disruptors. And a lot of these bacteria that grows in this agar, like this one, it's a tube, but with the agar inside the tube, where we get a single colony from that plate, and we stick that single colony inside this tube, right? And then you take a look after 24 hours, and you see this space in the agar. This is gas production. Why this is so important? Why it's so important to know that these disruptors produce gas? So let's remember that, and then you will see that later, that one of the diagnostic tests that we apply for SIBO, besides culturing of the small bowel aspirates, is breath test, right? Why we do breath test? Because all of these microbes, mostly associated with SIBO, they produce gas. And you can see that in vitro. Look at the amount of gas that a single colony produces after 24 hours. They produce hydrogen, they produce CO2, some of them produce H2S. Remember that I told you that that black spot in the colony is important? That black spot in the colony means H2S production as well. It's a marker of H2S. Not only that, let me go back. Not only that, even here, this is a black color in the agar, over here as well, and some points depending on the spot that you get in this test. All this black color means a positivity for H2S, right? So then with this range of tests, we can tell if the microbe can metabolize some types of carbohydrate and can produce gas, which type of gas. So if it's black, it's H2S. If it's transparent, it's hydrogen mostly and CO2, right? We can also check if these microbes have motility. This is important. Most of the pathogens, they are able to walk through the gut. So they can walk through the mucus layer, they can walk through to the cell layers. They do that. They have the capability to do that, right? And then we can see that on the agar as well. So usually when we stick the colony in the agar like this, and you see that the bacteria is going over the sides of the tube, this means that the bacteria has motility, right? So with this and many other tests that we do in the lab, we can tell the specific species that was involved or overgoing samples. In this case, that we do in the lab is mostly the reimagined samples, right, from patients coming from upper endoscopy. And the other thing that we do as well, not exactly for research purpose, but again for clinical purpose, we have other ways, more advanced ways, to identify these microbes without doing all these biochemical tests. We still need to do the visual inspection. We still need to grow them as we reach single colonies, right? And after that, the only step that needs to be done is getting a single colony and running a protein analysis, a full protein analysis, where you can check the protein pattern coming from this colony. And you can give the identification of these microbes. And this is exactly what we do at Cedars-Sinai in the clinical lab, not on the research lab. So we use this type of technology to identify microbes. We still apply biochemical tests. It is very important, especially to know if they're producing gas, and which type of gas. I also want you to understand that even the same species of bacteria can behave different in a specific environment. So we have Escherichia coli that sometimes produce only hydrogen and CO2, But we also have Escherichia coli that eventually can produce H2S. And that cannot be checked with this type of technology, only with biochemical tests, right? And then the next step would be finally doing identification using DNA. So then we do DNA amplification, and we use specific assays where we can increase the amount of DNA in a specific sample using probes. And those probes are specific to species that I'm searching for, like this one. We have arrays of tests where we can check for several species at the same time. And this is something that we also do in the lab and especially apply in clinical labs as well. But all of this comes with a cost, a huge cost, right? Because it's not always perfect. One of the things is that it's not every microbe that we can grow in the laboratory. It's not every microbe that we can even see it on the microscope, right? And then let's imagine the microbiome to a more broad range perspective. We are not growing every single microbe present in a sample, just some of them. So this is the major challenge that we have in the clinic. So how we can actually expand our knowledge, and instead of studying single microbes, which are important as well, how we can study the whole microbiome altogether. And this is what we do at MAST. Right? So we, instead of studying single microbes, although we apply these techniques to diagnose SIBO and SIFO, we apply cutting-edge technologies, high-throughput technologies to study every single microbe in a sample. So we are not isolating one, we are isolating all of them at the same time. And how we do that? And then I will walk you through this process. So now we left the clinical perspective of this lecture and going a little bit more over the research perspective, although we have companies these days that already offer tests to check your microbiome in the clinic as well. So that's why by the end of this block, I want to show you a few reports, because if by the end of this block you know how to read these reports coming from these companies, you understood everything that we have been talking about right now. Right, so to a more broad range perspective, to a more research perspective, what we do. We collect the samples from the small bowel, from stool, from any other site that you want to study the microbiome. We have a mix of cells, mix of microbial cells, bacteria, we have fungi, we have potentially parasites, lots of viruses all together. What we do next, we extract their DNA, because the DNA is the code that every single microorganism have it, and that code is unique to them. So this is a bacteria, one example, and this is the DNA that every bacteria has it, right? So then what we do in the lab, we need to break down this portion that's not DNA, and we do that, and we isolate the DNA first. So this is the first step that we apply in the lab. We isolate this DNA, but not only for one bacteria. We isolate the DNA from all of them at the same time, all mixed in a single tube like this one. And then after we do that, we apply or we employ our cutting-edge technologies or sequencers, and we sequence every single DNA in a specific sample. And we can do that in several ways. So I will show you what we do and what we are actually more available to you in terms of research and what we have been doing now up to these days, right? So most of the microbiome studies, they use 16S sequencing to sequence all the DNA present in a sample, specifically bacterial DNA. Now let's talk more about bacteria specifically. And why we do that? So although, like I said, every single bacteria has its own DNA that is specific to it, they have specific regions in this DNA. Let's suppose this is a DNA from the 16S gene of a bacteria. They have specific regions, the white ones. They are exactly the same between all bacteria. And then they have the yellow parts, where these yellow parts are the ones that is different between species. So then when we sequence all this DNA together, we can design probes that these red marks over here where they will cover a little bit of the white part. So I am amplifying my DNA from every single bacteria. That's the guarantee that every single bacteria is going to be part of my pool. But I'm also amplifying the yellow part, where it will bring me information specifically about species identification. And we do that, right? And then we do something very similar to fungi, but instead of using 16S, we use another component of their DNA that we call ITS. But it's basically the same. In a single sample, we amplified specific portions of the DNA that are common to all fungi and some specific spaces in this DNA that are specific to species as well. And then what we do after we sequence all of them, we run a lot of bioinformatic pipelines. We bring all these samples, all this information to the lab, to our computers, and we start to apply different pipelines for bacteria identification. And we have two goals mostly for that. We want to check specifically for diversity, and Dr. Mathur already mentioned that diversity is an important feature of the microbiome. We want to know how diverse is the small bowel microbiome, how diverse are the stool microbiome. And the second thing that we do is profiling. Right? So then I will show you a little bit how we do it so you understand a little bit of these terms, because this is something that usually appears in the reports from these companies that provide these microbiome studies in stool samples. So diversity means that after I sequence the DNA, I ran all the pipelines to identify this DNA, match them with a database, identify the microbes that are present in a sample. I want to know how many types of microbes exist in a sample, and this is diversity. So each of these ones over here, the red ones, the purple, the green, they represent one type of microbe. So then the diversity of this environment, in this case the gut, right, is measured by checking how many different microbes I have in a specific sample, right? And then let's suppose that in this sample, I will consider this diversity at this specific point. Now, if I remove one microbe, so let's pay attention on the red one. If we remove the red one, this environment does not have this red microbe anymore. So the diversity decreases. And because it decreased, because now it lacks that red microbe that was present before, now our diversity is over here. There is a decrease in diversity. And overall, what we want for a healthy microbiome is a balanced diversity or higher diversity, which means that you have a different type of microbes collaborating balanced together, working all together to maintain your body, to maintain your basic functions for digestion, for immunity, right? But then what happens if we substitute that red microbe by a green one? Our diversity is back, right? Then we can compare the sample 1 that had the red one with the sample 3 that now have the green one. They have similar diversity, but they don't have the similar microbiome profile. Because the first one, it was the red one over there, and now our third one is the green one. So that's why the second most important step in microbiome study is the profiling as well. So not only checking diversity, right? We want to know exactly what is there. And then when we do that, the way that we do that is applying all those pipelines where then we organize this data. We organize these microbes by their types, if they are by their abundance as well. It's important to know how many we have of each one in a specific environment. So we separate them like this, but by bioinformatics, right? And then after separating them, we apply different platforms where we can actually classify these microbes. Okay, these ones are Escherichia coli, they are from the family Enterobacteriaceae, they are from the phylum Proteobacteria. Okay, these ones are Staphylococcus, so they have a specific family, they have a specific phylum. So we do that, and because we know this information about all of these microbes, and they are present in a specific database, we can now understand the reports that we get. right? The charts that we get representing this microbiome. So basically, this representation of the small bowel microbiome that Dr. Mathur showed to you is just what I just mentioned to you. It's separating these microbes by different types. So let's suppose this green one might be this green one over here, and also take into consideration how much they take from a specific sample. From this microbiome, the duodenum, this case, the green microbe over here takes almost 56% of all microbiome present in that specific site, right? And then you check the blue one, the green one. Let's check who is the green one. This one, the first one, was from the Streptococcaceae family. And then we have a second one that is the light blue one, that is from the Veilloninaceae family, right? And then I can see who they are, and I can see how much they take of this microbiome. The report sometimes comes to you as a pie chart, on the other hand. So let's understand these pie charts as well, because this is a just different way that we organize the microbiome data. So again, we separate those microbes based on who they are and how we can classify them, and we organize them in a pie chart, taking into consideration their taxonomic classification, right? So in this type of pie chart, we have information about the phylum level, we have information about the order, the class level, order level, family, genus, and species as well. And this is exactly what comes in your reports when you send your samples for microbiome analysis. And that's why it's so important to understand. Meaning that if I'm checking for a microbe over here, let's talk about Klebsiella, the one involved in SIBO. So Klebsiella is right here. Klebsiella pneumonic, that is the species, that comes from the genus Klebsiella, all the way to the family Enterobacteriaceae from the phylum Protobacteria, right? So this is how we organize the microbiome data. And this profile is unique to every single person, to every single microenvironment. But we can also plot this profile in a specific graph like this that gives information about how your microbiome is similar to other microbiomes. So if my microbiome, let's take into consideration the gastrointestinal microbiome over here. If I am this dot over here, if someone has a microbiome very similar to mine, that person dot is going to be over here as well. So this graph actually shows how the microbiomes are similar or different. And this is exactly what you're going to see in your reports. So I'm not advertising any company, but I want to show a little bit of this report so you understand. But the first page in the report for microbiome analysis on the clinical part, although we don't use that for diagnostic purpose, right? Just mostly for research, is showing how the microbiome looks like in terms of diversity, in terms of profile. It compares your microbiome to the diversity of the overall population, saying, look, your microbiome is over here, and this is the overall population microbial diversity. Also compare how similar is your microbiome to the overall population. Your microbiome is over here, the profile, and this is the whole US population. This is fecal microbiome analysis. They also give you these pie charts. They also show you what is represented in your sample at specific taxonomic level, at phylum level. This is your microbiome, and this is the population microbiome in the United States. And then they show you what is the percentage of each of these phyla in your sample compared to the entire population. So this is how you need to see through these reports. And they do that even at a more deep taxonomic level, such as family level. But the basic is the same. They compare the abundance of a specific family in your microbiome to the population. And we can extend that to many other types of microorganisms as well. We can do that for fungi, for parasites, for viruses. But at the end of the day, it's always the same information that we are combining all together. We are searching for a specific species, and we are understanding how this microbiome collaborate between them, right? And this is what we do right now. Instead of you only checking bacterial composition of the small bowel microbiome, we search for other types of microbes there as well. Again, remember, when we extract the DNA, we extract the DNA from all of them, not only from one microbe. And we can also employ different types of cutting-edge technologies to give us information about all of them at the same time. But as compared to any other test, we also have challenges. All of this is only possible if we have databases where these databases provide us the information of the DNA information of these microbes. Without having this database, we don't know what we are doing, right? So we isolate the DNA, we sequence the DNA, but we have nothing to compare. If we have nothing to compare, we cannot identify these microbes. So this is one of the major challenge in terms of high throughput technology, and also a lack perhaps of how this microbiome should be reported and analyzed. So that's why sometimes it's so confused to see all these manuscripts and papers showing microbiome analysis from one population, from another population. Sometimes they use different pipelines, and you don't know how to compare them. But it's something that we are working on, and that's going to change in a very short period of time. But what I want you to take with you today is that although we know that we have imperfections in how we access the microbiome, we are pretty good already, right? So we know how to identify these microbes, and we know how to report them to you. And this is what is important to us right now. Thank you very much.